The fight against cancer is one of modern medicine's most enduring challenges. For decades, the path for a newly diagnosed patient has been fraught with difficult choices, often involving harsh treatments like chemotherapy and radiation that, take a heavy toll on the body. Scientists and doctors have long dreamed of a different approach, a therapy that is not only powerful but also precise, one that can hunt down cancer cells with surgical accuracy while leaving healthy tissues untouched. The goal has always been to create treatments that are smarter, faster, and most importantly, safer for the very people they are designed to save, turning a grueling battle into a more manageable journey toward recovery. Now, on this day, October 10th, 2025, we are witnessing a monumental leap toward that dream. Researchers from two of the world's most prestigious institutions, MIT and Harvard Medical School, have just unveiled a groundbreaking innovation published in the journal Nature Communications. They have engineered a special type of immune cell, a super soldier that can be described as invisible. These are not your typical immune cells. They are called CAR-NK cells, and they have been meticulously designed to perform two incredible feats simultaneously. They seek and destroy aggressive cancer cells while expertly hiding from the patient's own immune system, preventing rejection. To understand this breakthrough, we first need to appreciate the body's natural defenses. We all have immune cells called natural killer, or NK cells. Think of them as the vigilant sentinels of our system, constantly patrolling for signs of trouble like viruses or developing tumors. They are incredibly effective but can sometimes be overwhelmed by advanced cancer. The core challenge for scientists has been how to amplify their power. For years, the focus has been on another type of cell, the T cell, but this new research brings the spotlight squarely onto the unique potential of these remarkable NK cells, promising a new frontier in immunotherapy. The biggest hurdle in using cells from a healthy donor to treat a patient has always been immune rejection. When a patient's body receives cells from someone else, its immune system typically recognizes them as foreign invaders and mounts a fierce attack, destroying the therapeutic cells before they can even begin their work. This fundamental problem has limited the development of universal, off-the-shelf cancer therapies. But the brilliant minds at MIT and Harvard have engineered a solution that effectively cloaks these donor NK cells, rendering them invisible to the patient's defenses and allowing them to carry out their cancer-killing mission unimpeded. The genius behind these invisible cells lies in an elegant and highly efficient engineering process. The research team, led by senior authors Jianzhu Chen of MIT and Rizwan Romi of Harvard, developed a single powerful DNA construct that can deliver multiple genetic commands to a donor NK cell all at once. This is a significant advance from previous methods that required several complex and time-consuming steps. By packaging all the necessary instructions into one neat genetic bundle, they have created a streamlined system for mass-producing these sophisticated cellular warriors, making the therapy scalable and accessible. This all-in-one construct performs several critical functions to transform a standard NK cell into an elite cancer-fighting machine. First, it uses a tool called short-interfering RNA, or siRNA, to silence the genes that produce specific proteins on the cell's surface. These proteins, known as HLA class I, are like identification badges that the patient's immune system checks. By removing these badges, the engineered NK cells can move through the body like ghosts, completely evading detection and subsequent destruction by the host's T cells. This is the key to creating a truly universal donor cell therapy. Next, the DNA construct equips the NK cell with a chimeric antigen receptor, or CAR. This is the cell's targeting system. In this particular study, the CAR was designed to lock onto a protein called CD19, which is commonly found on the surface of cancerous B cells in patients with lymphoma. This receptor acts like a heat-seeking missile, guiding the CAR-NK cell directly to its target and ensuring it attacks only the cancer cells. This precision is what makes the therapy so effective, as it focuses the cell's destructive power exactly where it is needed most, minimizing collateral damage. Finally, to make these cells even more robust, the construct instructs them to express other proteins that enhance their lethality and help them survive in the hostile tumor environment. One such protein, PDL1, acts as a shield, protecting the CAR-NK cell from the cancer's own defense mechanisms. This multi-pronged approach, invisibility, precise targeting, and enhanced self-defense, results in a therapy that is not only safer by avoiding rejection, but also significantly more powerful, creating a cell that is purpose-built to conquer cancer from within the patient's body. A brilliant idea in a laboratory is one thing, but demonstrating its effectiveness is what truly matters for patients. To test their creation, the researchers conducted a compelling series of experiments using humanized mice specially bred to have a human-like immune system. These mice were first injected with aggressive human lymphoma cells, a cancer that spreads rapidly if left unchecked. The stage was set for a clear and definitive test. Could these engineered CAR-NK cells stop the cancer in its tracks, where other treatments might fail? The results were nothing short of astonishing, and provided a powerful glimpse into the future. The mice were divided into groups. One group received the newly engineered invisible CAR-NK cells. Another group, serving as a control, received NK cells that were either unmodified or had only been given the cancer-targeting CAR gene but not the invisibility cloak. The difference in outcomes was stark and immediate. In the mice treated with the fully engineered cells, the therapeutic CAR-NK cells persisted and thrived for at least 3 weeks. During this time, they hunted down and almost completely eradicated the lymphoma, leading to a dramatic regression of the disease. Meanwhile, the story for the control groups was tragically different. In the mice that received the unmodified or partially modified NK cells, the host immune system did exactly what it was expected to do. It identified the donor cells as foreign and swiftly eliminated them within 2 weeks. Without their therapeutic protectors, these mice saw their cancer spread uncontrollably. This direct comparison powerfully demonstrated that the genetic invisibility cloak was not just an advantage, it was absolutely essential for the treatment's success and the survival of the host. Beyond just efficacy, the study delivered more fantastic news on the safety front. One of the most feared side effects of other immunotherapies, particularly CAR T-cell treatments, is something called cytokine release syndrome, or CRS. This is a massive inflammatory response that can be life-threatening. Remarkably, the engineered CAR-NK cells were found to be significantly less likely to trigger this dangerous condition. This finding suggests that the new therapy could offer a much safer alternative, providing patients with a powerful weapon against cancer without the severe risks associated with existing treatments. The publication of this research has, unsurprisingly, generated a wave of excitement and optimism across the patient community. Already, people from around the world diagnosed with various forms of cancer are reaching out, inquiring about the possibility of joining future clinical trials. This immediate and passionate response underscores the urgent need for better, more accessible treatments. While it is important to note that this therapy targeted a specific protein, CD19, which affects both cancerous and healthy B cells, the promise of an off-the-shelf solution that can be administered immediately after diagnosis is a game-changer. The real beauty of this technology lies in its versatility. The one-step engineering platform is not limited to lymphoma. The DNA construct can be readily modified to equip CAR-NK cells with different targeting systems, enabling them to hunt down other types of cancer. By simply swapping out the CAR design, scientists could direct these invisible assassins to attack antigens found on solid tumors like prostate, lung, or pancreatic cancer. This adaptability opens the door to a future where a whole arsenal of ready-to-use cellular therapies is available to treat a wide spectrum of malignancies. Looking ahead, the road from the laboratory to the clinic is now being paved. The researchers are already planning to initiate human clinical trials in collaboration with the world-renowned Dana-Farber Cancer Institute. This is the critical next step in validating the safety and efficacy of the treatment in people. If these trials are successful, it could fundamentally reshape the landscape of cancer care, offering hope to countless patients who currently have limited options. The potential impact is so profound that the team is also exploring its use for other conditions, including autoimmune disorders, like lupus. For every person whose life has been touched by cancer, today marks a moment of profound hope. This breakthrough is more than just a scientific achievement. It is a beacon, illuminating a future where cancer treatment is not a punishing ordeal, but a precise, manageable, and ultimately curable process. While there is still work to be done, the creation of these invisible immune cells represents a giant stride forward. It is a testament to human ingenuity, and a promise that we are getting ever closer to a day when a cancer diagnosis is no longer a source of fear, but the beginning of a swift journey to healing.